So, our next question. Can goji berries be used to treat Alzheimer's patients? We hope to get the answer from our next speaker, Albert Kim from the United States. Welcome up on the stage.
Hi, my name is Albert Kim, and my project proposed a potential treatment for Alzheimer's disease using the HTB11 human neuroblastoma cell line, and I demonstrate the potential effects of an environmental toxin called 4-nonylphenol or 4-NP in relation to this novel treatment. Now, what is Alzheimer's disease, or AD? AD is a chronic disease that causes memory loss and dysfunction, and it is characterized by toxic amyloid deposits in the brain. Now, what is 4-nonylphenol or 4-NP? 4-NP is a toxic daughter molecule of nonylphenol phosphates. Nonylphenol phosphates are used as surfactants to produce various liquid detergents, cosmetics, paints, pesticides, etc. However, under anaerobic conditions, such as those found in wastewater treatment plants, nonylphenol phosphates biodegrade into the environmental toxin 4-NP. Also, 4-NP has been shown to be a major model for Alzheimer's disease due to its ability to mimic some of the effects, such as increased oxidative stress. So it's been surmised that antioxidants can be a potential treatment for Alzheimer's disease because of their ability to target and reverse factors that are directly involved, such as increased oxidative stress. Therefore, Lysine barbarum, or goji berry extract, presents itself as a potential treatment as it is the world's current strongest naturally occurring antioxidant. Now, moving on to the 24 genes that were investigated in this project, I clustered them into 3 groups: AD progression, oxidative stress, mitochondrial dysfunction, and I further clustered into genes that promote and reduce toxic amyloid deposits, and genes that promote and reduce oxidative stress. So the purpose of this study was to investigate the effects of lysine barbarum on stress response mechanisms induced by the environmental toxin 4-NP, a major model for Alzheimer's disease, and it was hypothesized that lysine barbarum would reverse all these harmful effects at the molecular level. So at the center of my project was a state-of-the-art technology DNA microarray, every step of which I performed. So I started off with the HTB11 human neuroblastoma cell line, and I split my cells into 8 groups. Control, goji berry extract or lysine barbarum at 2 separate concentrations, 4-NP at 1, and most importantly, the last 4, 2 24-hour pretreatments of lysine barbarum at 2 separate concentrations, and then the addition of 4-NP, and 2 24-hour pretreatments of 4-NP, and then the addition of lysine barbarum at 2 separate concentrations. After a 48-hour treatment phase, the mRNAs were extracted from these cells and transcribed their corresponding cDNAs via RT or reverse transcription. Afterwards, the cDNAs were applied to the DNA microarray chips and scanned for detection via chemiluminescence. Afterwards, the data was analyzed via Spotfire software, and to make sure that all the data up here is correct, I conducted a validation of the microarray through an RT-PCR for the base 1 gene, which is selected at random, and reference gene beta-actin. Afterwards, a statistical analysis via ANOVA test or analysis of variance, followed by post-hoc Chaffee with p less than 0.05 considered significant, was conducted. So instead of throwing a blob of data at you guys, I created diagram A to summarize the findings. So for the genes that support toxic amyloid deposition, 4-nonylphenol upregulated it, lysine barbarum downregulated, and we see the opposite effects for the genes that support the clearance of toxic amyloid deposits. For mitochondrial function genes, 4-NP downregulated, goji berry extract upregulated. For our oxidative stress promoter, 4-NP upregulated and lysine barbarum downregulated, and of course the opposite effect occurred for the oxidative stress reducers. So whether lysine barbarum was added before or after the treatment of 4-NP, we see that the data is constantly skewed towards the control. And this suggests 2 very important things. 1, that lysine barbarum is trying to bring the cell back into equilibrium or homeostasis. And 2, that lysine barbarum may not only act as a potential repairing mechanism against Alzheimer's disease, but also a potential protective mechanism. Lastly, I'd like to add that all these significant effects occur at concentrations far below the current EPA limit of 3,100 nanomolars. I show that these significant effects occur at 1,000 nanomolars, or 1 microgram per microliter. So a limitation of this project was that I investigate the effects of 4-NP for only 48 hours due to time restraints and practicality. However, in the real world, we're exposed to foreign peat for daily years and decades before effects actually surface. So in the future, I like to run tests potentially up to a couple months. Also in the future, I like to run protein assays or Western blots and ELISAs to make sure that all these changes at the genetic level are actually translating to viable proteins. Thank you for your time, and if you have any questions, come meet me at my poster board.
Thank you, Albert. So please share with me and with the audience, how would you describe a typical Swede?
A typical Swede?
Yes.
I think a typical Swede is very shy, pretty cool, and respectful. I understand you guys like your personal space, especially in the public transportation areas. And almost everything in your house is made by IKEA.
But maybe that's well, thank you, Albert. Thank you.
Thank you.
So our final participant for today is Peter Gaidaro, who has traveled here from Bulgaria